First, let me give you a little bit of introduction how I entered this research field. When I was at college, I have no idea what I will do in the future. During my sophomore year, I joined a research lab working on plants. They isolated active components from plants and applied them to treat high blood pressure. Although I didn't make anything significant for the project, this drug discovery process really inspired me. Ever since then, I have been passionate about drug discovery and development. Later on, I learned drug discovery and development has been considered as one of the most complicated processes in both industry and academia. So after graduation, in order to expand my knowledge and strengthen my technical skills, I obtained a master's degree in organic chemistry, a PhD degree in pharmaceutical sciences, and a postdoctoral training in chemical engineering. It took me over 10 years to complete all these trainings and studies, much longer than I expected when I was a sophomore student. But finally, I got there, I'm ready. I joined OSU in 2014. I set up my own lab in the College of Pharmacy and started my independent journey for drug discovery and development. So in the following talk, I would like to share with you one of our projects for the treatment of hepatitis B using nanotechnology developed in my lab. Hepatitis B is caused by hepatitis B virus, abbreviated as HBV. HBV mainly infects human liver, which is transmitted through blood or body fluids from infected patients. Hepatitis B is a worldwide pandemic and a severe global health burden. An estimated over 350 million people are chronically infected, and over 70,000 people die every year due to HBV-related serious complications, such as liver cancer or liver failure. Although the incidence rate is low in the US, still 1 out of 20 Americans have been infected by HBV. How HBV is treated now? One option is to boost the immune system for the patients using protein drugs. This approach can solve the problem for about 15 to 20% of patients. For the rest 80% of patients, they don't respond well to these protein drugs. They have to take antiviral drugs lifelong. However, these drugs only inhibit viral infection, but not cure hepatitis B, because HBV virus persists in the form of HBV DNA. This DNA keep producing viral proteins, which are the cause of the serious complications. So in order to address this issue and achieve a cure for hepatitis B, new therapeutic strategies have to be developed. One strategy is to learn from the nature. How does bacteria counteract viral infections? Bacteria developed a very smart strategy. They have a system called CRISPR. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a long name. You don't need to remember that. I cannot remember either. So this is generally an immune system in the bacteria. There's an important protein called Cas9, CRISPR-associated protein 9. How does this system work? This is a simplified illustration of the process. When virus invades bacteria triggers this CRISPR system, which is composed of a guide RNA and a Cas9 protein. Guide RNA identifies the viral DNA. Cas9, like a scissors, cut the viral DNA into pieces. Consequently, bacteria is able to get rid of viral infection. Very smart strategy. Does human cell produce this CRISPR system? No, we don't have this system. Could we utilize this system to work against the viral infections? Maybe. There are several challenges. One of the key challenges is to deliver the CRISPR system into the infected cells. Because the CRISPR system is not stable, after injection, the system will be degraded within minutes in the human blood. So we really need a delivery vehicle. Imagine we are truck drivers. we need to load the CRISPR system, transport them into the infected cells, and then this system can achieve the function and eliminate viral infections. So my lab is working on new nanotechnology which can serve as delivery vehicles. One technology is called lipid-like nanoparticles. We synthesize lipids, we formulated them into very nice nanoparticles. These particles are like mini trucks, which can deliver all kinds of therapeutic cargoes. So in this study, we are wondering if we can deliver the CRISPR system. We tried all kinds of formulations and conditions. Finally, we were able to load over 99% of the guide RNA and the Cas9 into those nanoparticles. This illustrates the overall delivery process. Nanoparticles protect the CRISPR system in the bloodstream after injection. And then they bring the system into infected cells. In those cells, the CRISPR system are unloaded. Guide RNA identifies the HBV DNA, and the Cas9 cuts the viral DNA into small pieces. This process doesn't affect the human DNA. So the cells are very happy to get rid of viral infection, and it's healthy. To test this design, we established a mouse HBV model. We injected the nanoparticles intravenously in the mice, and then we measured the HBV viral load. From this figure, we can see clearly nanoparticles dramatically reduced the viral loads compared with the control groups. Now we're wondering why you want to use nanoparticles. What's the advantages? Previously, people have reported virus to deliver the CRISPR system. However, our virus may induce immune response in the human patients, and also they may lead to other potential toxicity and side effects. These nanoparticles possess several advantages. They are very specific to the liver. They can be metabolized by the human cells, reducing all the potential toxicity and the side effects. So with the promising results, in the future, we plan to combine antiviral drugs and the nanoparticles. Antiviral drugs inhibit viral infection. Nanoparticles eliminate HBV DNA in the infected cells. Through this combination therapy, we anticipate achieving a cure for hepatitis B. So with that, I would like to thank my lab members. This is a group of talented researchers, including postdocs, graduate students, undergraduate students, and high school students. Our goal is to translate our nanotechnology into more effective treatments for all kinds of the viral infections, such as hepatitis, HIV, flu, and many other types. Thank you very much for your attention.